Great. Hello, everyone. My name's Katie, and I'm from the University of Washington, Doug Fowler's lab. And I first want to thank the organizers for the opportunity to present today. So today I'll be talking about variant in situ sequencing to visually phenotype thousands of PTEN variants. So I think a lot of the talks so far have really highlighted the great opportunities that are presented by mutational scanning data. And I think as this approach becomes more mature, a lot of us are looking to move beyond one-dimensional readouts and attempt to measure multiple things at a time. And I think this becomes especially important when we're looking at more complex phenotypes that we might not be able to read out through survival, for example. And in addition, I think we've been able to get a lot of great data using a variety of systems, but I think we also want to move beyond non-human or cancer cell lines and attempt to look at specific cell models and cell types. So the approach that I'm going to be presenting on today to kind of address some of these issues is using an optical phenotyping approach. And some of you may remember this project from a talk by a former lab member named Shriram, who presented this meeting last year on our project looking at LMNA using this approach. So we looked at 2,000 LMNA missense variants in U2S cells that had had LMNA knocked out. And so the image shown here is a library of these LMNA variants. And as you can see, we can measure nuclear morphology using a DAPI stain. We tagged the LMNA variant with GFP, so we can see its localization within the cell. And then we're also able to measure other generic morphology of the cell, including actin, as well as using a mitochondrial RNA FISH probe. So then the obvious question that we want to answer is which of these cells corresponds with which variant? So in order to do that, that's where in situ sequencing comes in. And so one of the key things that we're doing with this approach is using a circular RNA barcode. And what that does is it enables high expression levels of the barcode compared to a linear transcript. And so the first step is to do reverse transcription, and then we hybridize a padlock probe to either side of the barcode. We copy in the barcode sequence, and then we ligate it close so that we have a circular template that we can then perform rolling circle amplification to copy the barcode numerous times. Once we've done that, we can proceed with the in situ sequencing. So we will incorporate one fluorescent base, image that. Each channel corresponds to one of the nucleobases. And then we can cleave the fluorophore, incorporate the next base, and in that manner, we can sequence the barcode. And so in that way, we're able to match the variants to the phenotype images shown on the left here. And we're able to do that using long-read sequencing to match the barcodes to the variants. So in a typical experiment, we can collect around 4 million cells, and we use a pipeline called StarCall, developed by Nick in the Fowler lab. And then we're able to use an analysis called Cell Profiler to calculate features for each of the cells. So what that looks like is a table where each of the rows is a cell, and then it has an associated barcode, which we map to the variant. And each of the features we can calculate a wide variety of features, including cell size, nuclear size, intensity of each channel, correlation between channels, and then other features such as granularity. So what we do is we then aggregate all the cells for a given variant and attempt to understand how the different cell images change at a variant level. And so shown here on the left is a UMAP where each dot on that UMAP is a variant. And the distance between dots on that UMAP represent their distance in morphology space. So what you might be able to think about that as is the difference in how the cell images appear. But the obvious question we want to be able to answer is, how are those images different, and how does that relate to the fundamental biology we're trying to study here? So one thing that you might notice is that in the gray and the yellow-green color here, those correspond with clusters 8 and 9. And one of the features that we see is increased nuclear granularity. And this corresponds well with previously published data, which had shown an increase in aggregation for variants showing up in this area of the UMAP, where more yellow color corresponds with a higher percentage of aggregation for the single variant testing that they did. So this was really promising, and we were able to learn a lot about LMNA variants from this analysis. But we wanted to move into a different cell line and not just use U2S cells. And one of the reasons that we wanted to do that was because we wanted to study the PTEN gene. One of the reasons that we were interested in PTEN is because variants can cause different diseases. So germline variants in PTEN can cause autism and also a tumor syndrome. It's also involved in somatic cancer. And so in order to do this, we wanted to study in iPS cells and then also NGN2 neurons. But to do that, we needed to create a method of not having our transgenes silenced. So that was the first step that we had to do. And for that effort, we designed this construct here, which allows us to express this transgene and not see silencing. So on the bottom left, you can see over 14 days, shown in blue is our construct, and it continues to express over time. And then the red line shows a lentivirally incorporated construct where we began to see silencing over the 14 days. And the other benefit of this is that you can see using the circular RNA, we're able to get many more dots per cell. And what that allows us to do is to map a higher percentage of cells to our library. So now that we can express a transgene over time, we can now look at the PTEN gene again. And so PTEN is a phosphatase, and so we designed a small mutagenesis library around the catalytic P loop. So that was from positions 112 to 172. And so the assays that we performed were both in iPS cells and then also in NGN2 neurons. So we took iPS PTEN knockout cells, put in our variant library using piggyback, and then did the assay in that cell type, as well as using docs to induce NGN2 neurons, and then performing VIS-seq in neurons as well. And one thing that I want to highlight here is we used some similar markers from before, but we also incorporated an anti-phospho-AKT antibody, which allowed us to measure the phosphatase activity of PTEN. So this allowed us to get multiple variant effect maps from a single assay of both abundance, phosphatase activity, and localization by looking at PTEN abundance, phospho-AKT intensity, and as well as the correlation between DAPI and PTEN. So one obvious question was, how does this correlate with previous results? And actually, Taylor Miguel collected some of this data. So fortunately, it did look like a pretty good correlation for the phosphatase score. And then the abundance data also showed a good correlation. So that was encouraging that our data was matching decently with previously acquired data. So then one thing that we also wanted to look at was which of these features were showing up for variants, like how common were these changes. And we noticed that of the missense variants we looked at, only 192 of them did not have perturbations in any of these three features across the two cell types. So it does seem like variants were commonly causing these changes in these features. And also of interest to us was that the DAPI PTEN correlation feature that I mentioned was commonly perturbed, especially in iPS cells. And so we obviously knew going in that abundance and phosphatase activity would be important, but we were surprised by the prevalence of this phenotype, and I think that's a benefit of like an optical readout in this case. So looking at synonymous versus nonsense variants on the UMAP, and once again, each point here is a variant, and distance between the variants indicates their distance in morphology space and the difference between the cell images. So we got good separation between nonsense and synonymous variants, and nonsense here is shown in red and synonymous in green. And then we wanted to see how this looked for whether or not we could explain some of this with abundance and phospho-AKT. And so, as you can see, it seems like one of the primary features of this UMAP, right, is like these two large clusters, especially evident in the neuron data. And it seems like a large part of that is described by the phosphatase activity. But as Kristen mentioned earlier, we do see some variants where there's not a decrease in abundance, but there is a decrease in activity. So that was helped explain the structure of the UMAP. And then we wanted to look at pathogenic and benign variants. And we saw a good separation, especially for the iPS cells, with the likely benigns all clustering together, and the likely pathogenics and pathogenics largely clustering in other areas of the UMAP. So we then wanted to look deeper into this to see if we could say anything about the various disease types associated with PTEN. So there's multiple colors here. Blue showing nomad variants that aren't in ClinVar, so are likely to be associated with benign variants, and then ASD or developmental delay variants shown in beige, combination of PHTS and ASD variants shown in this darker red, somatic cancer in this lighter red, and then PHTS in this pink. And we see that many of the variants associated with either PHTS or somatic cancer are showing up in this left area of the UMAP, which makes sense given their published decrease in phosphatase activity. And then interestingly, the autism variants are less often there, which corresponds with previously published results that show that we don't see that same decrease in phosphatase activity for those variants. So we wanted to look at what other features might explain some of these autism variants, and one of the things that we looked at was the mislocalization score. And one thing that we found interesting was that it seemed that both the autism-only variants and then some of the patients who had both autism and PHTS, we saw a slight increase in the mislocalization score, which meant that the PTEN was more likely to be localized to the nucleus compared to both synonymous and then also the purely tumor-causing diseases. So then we wanted to see, more broadly, how is our data allowing us to compare between pathogenic and these different diseases. And so showing here 3 different ROC curves, where the first one is just ClinVar pathogenic versus likely path, then tumor syndrome, and then lastly ASD on the right here. And so one thing that I wanted to highlight here is that if you look at ClinVar in general, I guess across all 3, we saw a slight increase if you just compare it to abundance or phosphatase activity from the yeast data set. We saw an increase in ROC, but I think, for example, for PHTS, where phosphatase activity seems to be a pretty dominant determinant, the yeast fitness already does really well. It has a really strong AUC. Whereas for the autism variants, it seems that we get a bigger boost from having this additional data there to complement. And then lastly, I wanted to touch on variant effect predictors. So we saw a similar performance for Revel and Eve, but I'm just showing alpha missense here. And the impact score that I'm showing on the left is our attempt to look at a synthesis, I guess, of all of the feature data. So we're looking at the cosine distance of these feature vectors to try to evaluate how far a given variant is from synonymous variants. And what we see is pretty good correlation with the alpha missense score when we aggregate that data together. However, when we look at some of the individual features that I talked about earlier, we see a less good correlation, which isn't that surprising given that these predictors are not designed to predict individual features like these. So some future work is we hope to collect data for additional variants to have a larger sample size to understand this gene-disease relationship that I talked about, as well as collect single-cell RNA-seq data for LMNA variants to try to see how that compares to some of the visual phenotypes that we saw, as well as do some further investigation of the PTEN mislocalization for the ASD variants that I talked about. So hopefully the data I've showed today has helped convince you that VIS-seq is a powerful mutational scanning technology. It enables silencing-free optical pooled transgene assays in iPS cells and other differentiated cell types. And it also enables a multimodal readout of any imageable phenotype. Shown here, I talked about variant localization, but we can also do more broad cellular morphology features as well. Lastly, we're able to collect data from millions of cells per experiment at around 100-fold less cost than single-cell RNA-seq, at less than a tenth of a cent per cell. So this is why we think this technology could be really scalable and provide really valuable data for other genes as well. So I really want to thank everybody in the Fowler Lab, especially Doug for his mentorship, and Sriram for collecting the LMNA data, as well as leading a lot of the analysis for this project. Nick for developing the star call pipeline that allows us to analyze this data, and Abby for her work curating PTEN variants. So thank you, and I'd be happy to take any questions.